In part 1 pancreas, we have learned the exocrine function and anatomy of pancreas. Here, today we are going to learn about the endocrine function. That means endocrine function in the sense the specialized cell which is responsible for the secretion of the some hormones directly to the blood. Here the special cell is called as a islets of Langerhans. In human, pancreas contains around 1 to 2 millions of islets of Langerhans. Here these cells is responsible for the secretion of the different hormones. Here the endocrine part of the pancreas contain the 4 type of cells that secrete different hormones. These 4 cells, that is the alpha cells or A cells, it constitute about the 17% of the pancreatic islets cells and secrete glucagon. The beta cells or B cells, it constitute about 70% of pancreatic islet cells and secrete insulin. The next cell is delta or D cells. It constitute about 7% of pancreatic islet cells and secrete somatostatin. Then fourth cell, that is the F cells or PP cells, it constitute about 6% of pancreatic islet cells, secrete the pancreatic polypeptides. These are the important hormones secreted the islets of Langerhans present in the pancreas. Hormone is glucagon. Glucagon is a polypeptide hormone. It's having a molecular weight is 3485. Its half-life is 3 to 6 minutes. It is secreted from the alpha cells of pancreatic islets of Langerhans. This glucagon is also produced from the A cell present in the stomach and intestine. Here the secretion is occurred when the decreased blood level of glucose, some exercise, stress, some protein meals stimulate the secretion of glucagon. The somatostatins and insulin inhibit the glucagon secretion. Here some of the ketones and some of the fatty acids are also stimulate this glucagon secretion from the A cell of pancreatic islets of Langerhans. Then it is a hyperglycemic hormone, glucagon. Here that means hyperglycemic means that increase the blood glucose level. Here liver is the main target of this hormone action, because here the glucagon increase the hepatic glycogenolysis. Glycogenolysis means there is a breakdown of the glycogen to the glucose. But this glycogenolysis is not occur in the muscles. Gluconeogenesis is also increased by the glucagon. What is gluconeogenesis? The glucose is formed from non-carbohydrate sources like amino acids, some fats that may converted as a glucose. Here this happen in the liver by stimulating the enzyme responsible for the there is a conversion of a pyruvate into the phosphoenol pyruvate. and increasing the transport of amino acid to the liver, where amino acid is converted to glucose. So that is a process involved in the gluconeogenesis. But here the first that is already told to you, that is a pyruvate into the phosphoenol pyruvate. Here the glucagon that stimulate the pyruvate decarboxylase enzyme that convert the pyruvate to the oxaloacetate. Then oxaloacetate is by adding the phosphate group that forms a phosphoenol pyruvate. Okay, so that is the gluconeogenesis process. That is a step involved in the gluconeogenesis process. Here the glucagon is also increase the lipolysis and the ketogenesis. Lipolysis means the breakdown of the lipid into the free fatty acid. Thereby some ketone bodies are also formed. So that is called as a ketogenesis. It means the formation of the ketone bodies, beta hydroxybutyrate, acetoacetate, etc. These ketone bodies are formed. It stimulates, that is this glucagon is also stimulate the secretion of insulin, growth hormones and somatostatins. Here other actions of the glucagons are inhibit the secretion of the gastric juice, increase the secretion of the bile from the liver. That's about the glucagon. Now the next hormone secreted by the delta cells, that is a hormone is called as a somatostatin. It is a polypeptide hormone. It is also secreted from the stomach and the nerve ending of the brain. The half-life of the somatostatin is 2 to 4 minutes. Here the secretion of the somatostatin inhibited by, that is a pancreatic polypeptide hormone. The main action of the somatostatin means that is in pancreas on ions of Langerhans inhibit the alpha and beta cells, and also that is the inhibit the secretion of the both glucagons and insulin. That means somatostatin that inhibit the secretion of the glucagon and insulin. What is the action on GIT? It decreases the gastrointestinal motility and secretion of the gastrointestinal hormones like gastrin, cholecytokinin, etc. It is also inhibit the gallbladder constriction, thereby decrease the secretion of the bile. In the other action is brain. In the brain, inhibit the secretion of the growth hormones and thyroid stimulating hormone. Thyroid stimulating hormone is secreted from the anterior pituitary lobe, and it acts as sometime this there is a somatostatin acts as a neurotransmitter in the brain. The regulation of a somatostatin secretion. Usually somatostatin secretion is increased by the presence of glucose and amino acids and some cholecytokinin, and some in the case of the GIT, that is a stomach, GIT, gastrointestinal tract and stomach, that is increased secretion by the presence of a chyme containing glucose and protein. In the presence of the protein in the intestine, in the presence of chyme containing glucose, that increase the secretion of a somatostatin. The next hormone is pancreatic polypeptide. It is secreted from the F cells or PP cells in the ions of Langerhans of pancreas. Half-life of this hormone is 5 minutes. Then here the pancreatic polypeptide hormone is degraded and removed from the circulation through the kidney. Here the secretion, that is the secretion of the pancreatic polypeptide, that is meals containing the protein, and fasting, that is and exercise and acute hypoglycemia stimulate the secretion. Hypoglycemia means decrease the glucose level in the blood. But this condition, that means meals containing proteins and fasting and exercise and acute hypoglycemia that stimulate the pancreatic polypeptide. Here, the somatostatin and elevated blood glucose level inhibit the secretion of an pancreatic polypeptides. Uh, the main action of an polypeptide hormone, that is a pancreatic polypeptide hormone, that inhibit the pancreatic and biliary secretion. It delay the gastric emptying time. Its secretion is increased during the fasting, exercise and hypoglycemia, reduce the absorption of a food. That means pancreatic polypeptide reduce the absorption of food. It promote the secretion of the pancreatic digestive enzyme. These are the important action of an pancreatic polypeptide. Another important hormone secreted from the pancreas, that is from the aisles of Langerhans, that is insulin is secreted from the beta cells of a pancreatic aisles of Langerhans. Insulin is a polypeptide hormone with an 51 amino acid. Insulin has a two chain, that is the alpha chain and beta chain, which are linked by disulfide bond, that is a bridge. Here alpha chain containing 21 amino acid, beta chain containing 30 amino acid. The biological half-life of an insulin is only the 5 minutes. Then the main action of the insulin, that is already we learned, that is the hyperglycemic hormone is glucagon, but here insulin is a hypoglycemic hormone. Insulin favors the anabolism and cause the storage of carbohydrates, proteins and fats. The main target organs of insulins are liver, muscles, adipocytes in the adipose tissue. Insulin is an important hormone that regulates the blood glucose level. It is also control the carbohydrate metabolism. That's why any deviation, that means deficiency of an insulin that lead to some condition, that is a condition is called as a hyperglycemia. The hyperglycemia condition is called as a diabetes mellitus, that is increased glucose level in the blood. That means there is a normal random blood glucose level is 80 to 120 milligram per 100 ml, but that may be high. That means that condition is called as a diabetes mellitus. Okay, that we discuss later only. That's why that is insulin has an important action on a carbohydrate metabolism. Here there is insulin that increase the transport and uptake of glucose from blood to the cells by increasing the permeability of a cells. Sometimes some transporters, glucose transporters are also involved in the transport of a glucose from blood to the cells. So that means in the presence of insulin that increase the transport. That is here almost all the cell tissues and cells of our body that utilize the glucose, especially muscles, liver and adipocytes in the adipose tissue. Another important action of the insulin, that is these all actions coming under the carbohydrate metabolism, effect on carbohydrate metabolism. First what we discussed the same thing. Now that is second one is in the presence of insulin that promote the peripheral utilization of a glucose. That means in the presence of a presence of a insulin, glucose enter into the cells and immediately oxidized. That is a peripheral utilization of a glucose that promoted by insulin. Okay, thereby decrease the level of the glucose in the blood. Another important action means here in the liver, it stimulates the glucogenesis and inhibit the gluconeogenesis. And already we learned what is a glycogenesis means. Glycogenesis means the synthesis of a glycogen from the glucose that is occurred in the liver. Okay, glycogenesis is stimulated by the insulin, but the gluconeogenesis is inhibited by the presence of insulin. So gluconeogenesis means there is a glucose is formed from the non-carbohydrate source like a amino acid protein, that is the gluconeogenesis. The insulin, another important action means insulin that inhibit the glycogenolysis. That is the glycogenolysis means here the breakdown of the glycogen to glucose. That means once the glycogenolysis occur means what happen, glycogen is break down to the glucose, so the blood glucose level is increased. But here insulin that inhibit the glycogenolysis, the breakdown of the glycogen to the glucose is inhibited by the insulin. Then insulin is also promote the storage of glucose. Here the insulin that increase the glycogenesis, that means conversion of the glucose to the glycogen and stored in the muscles and liver, that is promote the storage of tissue. In this all way insulin that decrease the glucose level in the blood. Then the effect on protein metabolism, that means the second action. Already we learned how the insulin influence the carbohydrate metabolism. The second one is the effect of protein metabolism. Here insulin promote the protein synthesis and storage. That means insulin stimulate the transport of amino acid to the cells where they are used for the synthesis of a protein. As we know, there is a protein is nothing but the building block of the protein is nothing but the amino acid only. Insulin inhibit the catabolism of proteins. That means catabolism means the breakdown of the protein to amino acid. That means the insulin which hasten the synthesis of protein synthesis, but it inhibit the catabolism of a protein. Another effect, that means on fat metabolism, how insulin influence the fat metabolism. Usually as like how the insulin increase synthesis of protein, same way here the there is a synthesis of a fat is also stimulated by the insulin. It is also increase the storage of fat in the adipose tissue. That means in detail how this happen means, the first one is insulin promote the fatty acid synthesis in the liver from the excess glucose. That's why some of the that is obesity, some that is if you avoid the fatty meals, but you are not taking the glucose also, that will reduce the that is obesity, or some that is some type of the cholesterol. That means, but in the case if you avoid the fatty material, but if you take the more glucose, means what happen means here the glucose is converted to the excess glucose is converted to the fats. Okay, that is one of the reason. So during the that is if the cholesterol is increased means you just decrease the both, that is fatty materials as well as the glucose in the diet. Okay, so that is that's about the insulin. That means insulin promote the fatty synthesis in the liver from the excess of glucose. So the next one is here the transport of a fatty acid into the adipose tissue. Here that is achieved by the help of the lipoproteins. That means insulin facilitates the transport of fatty acid into the adipose tissue with the help of the lipoproteins. The next next step means the insulin inhibit the lipolysis. Lipolysis means the conversion of fats or lipids to the fatty acids. that is inhibited by the insulin. Next one is the storage of fats. The insulin promotes the storage of fat in the adipose tissue by inhibiting the enzyme which degrade the triglycerides. The next effect means the growth and development. How the insulin influence the or stimulate the influence the growth. That means usually insulin promote the growth of the body along with the growth hormones. Then another effect is the mineral metabolism. Usually insulin increase the entry of the potassium ion to the cells, thus decreasing the concentration of potassium in the extracellular fluid. Regulation of insulin secretion. Here regulation of insulin, that means insulin secretion is mainly depends upon the blood glucose level. In addition to the blood glucose level, that means sometimes the gastrointestinal hormone, that is hormone, some hormonal influence, that means some of the lipid contains an amino acid, that is stimulate the that is some nerve fibers is also cause the stimulation of secretion of insulin from the beta cells of ions of Langerhans. Then we can discuss that is a regulation, that is insulin secretion regulated by the three level. That is the first one is the substrate control level. The substrate control level means it depends upon the blood glucose level. When blood glucose level is decreased, that is a fall in the below or the normal, that is less than 70 milligram per 100 ml, then decrease the secretion of an insulin. But when the blood sugar level is become more than 70 milligram per 100 ml, the secretion of insulin increase manyfold. That means the substrate in this way the substrate level that control the insulin secretion. Here, in addition to the glucose, some amino acid level in the blood is also stimulate the insulin secretion. Next one is the hormonal control. Hormonal control means here the growth hormone, that is glucagon, cortisol, that is gastrin, secretin, cholecytokinin, etc. that stimulate the insulin secretion. Here the somatostatin inhibits the insulin secretion. And except the somatostatin, these other hormones like gastrin, secretin, cholecytokinin and gastrointestinal hormones like this hormone that stimulate the insulin secretion. The third control is a neural control. That means we know ions of Langerhans receive the both sympathetic and parasympathetic supply. Here the sympathetic stimulation, nerve stimulation suppress the insulin secretion, but the parasympathetic stimulation that increase the secretion of insulin. So that is the nervous controls. That's all about the action of the insulin. Now we learn already we discussed what are the hormones secreted by the ions of Langerhans, that is pancreatic polypeptides, somatostatin, insulin, glucagons. Their action is also we discussed. Thank you for watching this video.